If you could see me in person, I'd be blushing. Maybe the light's sort of obscuring the view here. Thank you so much for the invitation, especially from students. I always just love the students more. I don't mean to insult faculty, but I love the students in departments more than the faculty typically, because they're really the lifeblood of ingenuity. So my talk today, let me see if I can share my screen properly. My talk today, like Anya said, is about modeling phylogenetic, developing phylogenetic models for evolving interactions with an ecological context. I'm not a great multitasker, so I'll be quiet for a moment. There we go. And this talk is a little bit different from the types of talks I normally give, so just bear with me. I'm trying to give an overview of different themes that I've been working on. And the three research directions I'll present today won't have too much detail in terms of the math and the methods, but rather just want to give you a flavor for some of the types of issues that we grapple with when dealing with these types of models up here, phylogenetic models of evolving ecological interactions. And the main question is that I'm focusing on from the biological perspective is, how do phylogenetic lineages evolve as they interact with other lineages and with their environment? So that's really the framework I'm working within here. And I'm not sure if it's showing these faces on the side of my screen. Let me minimize it. There we go. And I'm just going to start off with some quotes from collaborators of mine. We'll talk about some of this work we did together in Viburnum in a moment towards the end. But I'd done some research previously with Michael Donoghue and Erica Edwards on a clade of flowering plants called Viburnum. And I did my postdoc work with Michael Donoghue. And what impressed me about that group is just the emphasis that they place on really knowing your system, really knowing a clade, knowing the biology, knowing where they live, knowing how they interact with the climate and other lineages and so forth. And when I was studying and when I was working on that topic with them, they repeatedly just drove home this point that, to understand how these things evolved in space, how they evolved over time, how they evolved ecologically, you just need to know specific details about the biological basis of the organism. And I just love quotes like this. In this case, we were able to integrate extensive field observations on leafing phenology to identify coordinated leaf senescence in particular as a parallel response to prolonged freezing during winter, and a possible mechanism underlying the repeated emergence of deciduous leafing habit in the temperate zone. I mean, there's a lot going on there biologically in that sentence, and it's through observation primarily. Now, I'll pick on them a little bit because and this is not unique to them, but it's unfortunate that many who study these organisms in such detail, when it comes time to actually reconstruct what's happening, they have to resort to some pretty crude models. So in a 2017 paper, they then went back to reconstruct certain ancestral ecological traits, such as being evergreen or deciduous, habitat types, the cold tolerances, and so forth. And to do these reconstructions, you have to rely on extremely generic models, such as Brownian motion models, Markov models, and so forth. And it's really dissatisfying because these models, because they're generic, they don't really tell you anything about what's happening specifically. They're essentially random walks. They're essentially blind to other factors that could steer traits to evolve in one direction versus another. And it really limits what you can say on one hand. On another hand, these Brownian motion models, these extremely simple models, they seem to be blind also to factors that can have concerted effects across multiple lineages based on where species lived in the past, what the local ecological conditions were in the past, and so forth. And here's a quote by Felsenstein, who in 1985 wrote a really influential paper, especially for evolutionary biologists and ecologists, that helped us think more clearly about what it means to talk about trait correlations and how might you sort of correct for correlations in traits caused by phylogeny. And in that paper, he introduced the notion of using Brownian motion to correct for these factors, but somehow Brownian motion kind of became a default model for thinking about trait evolution when you don't know what's going on. Now, if you start thinking about more sophisticated mechanisms for how certain types of ecological traits evolve, it becomes complicated very quick. So, I mentioned a moment ago that Brownian motion models are essentially just directionless random walks. This nice review paper by Harmon and others in 2019 sort of laid a framework for possible factors you might consider if you were thinking about ecological interactions between lineages and space over time and so on and so forth. And it's very easy to come up with a long list of possible factors that would contribute to how species evolved in sort of phenotypic space on the y-axis and over time on the x-axis. And it will be based on, say, interactions, what region they're in maroon or teal, and so on. So I just want to give you a sense of how complicated it can be. I don't think anybody's a stranger to that concept. Now, these simple models are really nice because of some of the assumptions they make. They generally assume that lineages evolve independently from one another. They assume that characters evolve independently of one another. They have extremely general mechanisms like the random walk mechanism. And this is wonderful because it means that you can efficiently do statistical inference under these models. But the reasons why you might forbid those interactions between lineages and characters is because it's often very difficult to justify exactly what those interactions look like. Having interactions with respect to characters and lineages creates a real inference problem. It means the state space explodes. There's more sort of combinations of ways that things interact that you have to account for. And fundamentally, it really breaks our simple marginalization algorithms, which is a fancy way of saying it destroys the way that the typical algorithms we use to probabilistically average over different histories. So what I'm going to focus on in this talk are sort of three different layers for how we might think about the ways that lineages evolve in an ecological context. I'll discuss one aspect where we're thinking about how lineages within a clade might compete with one another and co-evolve, how lineages between two clades might co-evolve, and how lineages might evolve within a set of biomes. So those are the three major aspects I'll focus on today. And the examples in particular will first be in Darwin's finches, looking at competitive exclusion with respect to shaping patterns of allopatry. Plant herbivore interactions between butterflies and angiosperms primarily, and how that might reflect on ecological network structure. And then lastly, how that plant group I mentioned early on, viburnum, evolved with respect to continentally sized regions and different major habitat types. And I'll touch on why this is a sort of difficult inference problem. Again, I'll try not to get into too many details for the math. So just think of this as something enjoyable to think about at the end of the day. So first, let's look at evolution of interactions within clades with Darwin's finches. Darwin's finches are birds. One very common pattern you see in birds is that they have some of the most striking patterns of allopatry among most living species, especially in South America. It's very common to find groups of closely related bird species that are almost entirely allopatric, which doesn't seem to be entirely by coincidence. Another interesting aspect of birds, and here are examples from a paper by Lack in 1947 and Diamond in 1972, is that you also often find characters that effectively displace one another in somehow. So for example, here on the left-hand side, when you have numerous different regions and you look at the bill lengths for different species, or for two different species, you find that in the regions where they overlap, the characters have essentially no overlap. So it appears that there's some type of character displacement going on, or potentially competitive exclusion. And on the right-hand side, you see for different sites across a mountain range, you find alternation in terms of which species is at a higher altitude and which species is at a lower altitude. And it's not entirely deterministic. There's some stochastic component. So in work with Ignacio Quintero, somewhat recently, we set out to better model how phenotypic traits and biogeographic ranges might co-evolve in some manner, and particularly how different competing lineages might affect the evolution of both of those traits. As part of this, we also tried to set it up in a framework where you could test to detect what types of biotic interactions are at play, and also to map these effects back onto a phylogeny somehow. So those are the three major goals for this study. So the basic problem here And I think most of the work we were building on was laid out nicely by Jonathan Drury and others, and Neusmer and Harmon in the mid 2010s. I'm not sure what the word for that is. But the basic problem is, suppose you have a phylogeny and you're thinking about what's happened back in time. So if you took a time slice at some point in the history of that phylogeny, you might have some number of species where each species is represented by a shape. And each species might have a trait, such as a +1 or a -1, to keep it simple. And they might occur on different regions. So we just have 3 islands here. So let's just call this the coevolutionary state for the entire system. And in this early work, especially the Drury et al. Paper, they laid out a framework where you could talk about the way that lineages might interact if they're in sympatry, if they share a particular region, for example. It might induce character displacement through competition, for instance. What we are interested in are these reciprocal effects. So not only that species, when they're in sympatry, can cause character displacement or divergence, you would also think that the biogeography of species themselves should be affected by what species with what traits co-occur or do not co-occur with you. So if you're a species with a +1 trait in the same region as another species with a +1 trait, maybe your rate of extirpation is higher. Or if you're a species with a +1 trait and you're dispersing into a new region that does not have a +1 trait there, maybe it's easier for you to disperse into that region than otherwise, right? So there's some type of reciprocity between biogeographic range and trait evolution. Now, again, I'm trying not to get into too many details, but what's tricky about this problem is that we want to model 2 processes simultaneously, and they both depend on each other. So let's call X of I a process that tracks the value of species I at time t. So just think about that for a moment. What trait does a species have at a given time? It's going to depend on its ancestral trait, and also the traits of all other species, just X without index I, and also all of the other ranges of all other species, big Y. Big Y down here is the biogeography of an individual species at time t, and it conditions on all the other traits in the system and all the other ranges in the system. So it's very high dimensional state space, this coevolutionary state. Now, for our model, what we're going to do is define 3 parameters. We call them omega x, omega 1, and omega 0. And depending on the value of these omega values, it determines, it will basically influence how different traits evolve, whether they tend to diverge or converge, depending on what other traits are in the region, say through competitive interactions or some type of facilitation. Or for the omega 1 and omega 0, these parameters will control whether rates of colonization for omega 1 or rates of extirpation for omega 0 increase or decrease depending on what traits are in the region. And I'll show you what I mean by those in a moment. But just as general context, I'm going to be using these 3 omega parameters to control how important the other lineages, traits, and ranges matter for trait and range evolution. So it's kind of a mouthful. Again, a lot of moving parts, but we'll try to walk through it somewhat slowly here. So let's first focus on omega x. So I said that is the parameter that controls how traits evolve in a biogeographic context. So this parameter omega x says that traits of sympatric species, when they're in the same region, can diverge or converge when omega x is less than 0, when it's negative, or when omega x is positive, respectively. And here, this plot, which is color-coded, kind of shows that behavior. So on the x-axis, we have time. Imagine this is evolutionary time going forward. And on the y-axis, we have x of t. Imagine this is the value of a trait for an individual lineage. And then just imagine that this lineage is exposed to some amount of some number of species with similar traits. So if a species is exposed to other lineages with similar traits and the value omega x is -1, that species would tend to diverge in trait from the other species. So you can see that this species goes this way, that species goes that way. If omega x is large, positive that is, the 2 traits will be drawn towards another, much like an Ornstein-Uhlenbeck process works, if that's a familiar term. So there are different processes that could potentially explain this, such as some type of environmental filtering or even admixture. But nonetheless, we have these two types of parameters, parameter values for omega x that could allow traits to diverge or be attracted based on who else is in the region with you and what traits they have. Now for omega 0 and omega 1, they both behave very similarly. So these two next slides will look similar in accordance to that. Let's first look at extirpation rates for omega 0. If omega 0 is positive, if it's greater than 0, it means that extirpation rates increase when sympatric species have similar traits. So imagine you're a species in a region and everybody else has the same trait as you for beak shape. It might mean it's harder for you to remain there as a lineage because of all the other competitors. So here on the x-axis, if all the other species in your region have similar traits, so the average distance in traits is 0, you would have a larger than average rate of extirpation up here in orange. 0, sorry, I think this is where 0 is for this particular plot, the black center point. Now, if omega 0 was negative, your extirpation rate, purple, would be, your extirpation rate would decrease as your traits became more similar. So it'd be easier for you to remain in the region given other species were in that region as well with similar traits. Now, as your traits become less similar, as the x-axis increases, the effect sort of converges to having no effect whatsoever. You're sufficiently different from everything else, so who cares who else is there? Now, colonization rate, like I said, these are two different slides. They use the same functional form. And they have the same interpretations for signs, basically. Except for, I reversed the order up here just because it's sort of intuitive to think of it that way. But in this case, if you have many similar traits, if you have many species with similar traits in the same region, the effects of competitive exclusion might be greater. And so therefore, it might be harder to enter a new region where other lineages with similar traits are. So I'm just giving you a sense of how these functions work and these relationships work. Now, what's complicated about this is that there are many possible places where species can live. There are many possible traits that species can have at any given time. And somehow we have to compute, we want to compute the probabilities for any of these traits and any of these regions evolving over time for the entire system, for all of the lineages in the system. And we can't use standard likelihood equations for that. So what we did is we used a data augmentation approach, which is essentially a technique for treating an evolutionary history as a very high dimensional parameter. So in the same way that if you're using Markov chain Monte Carlo, you can estimate things like divergence times, you can estimate things like species relationships, you can estimate things like rates of molecular substitution. We're going to do something similar like that, except we're going to encapsulate all of the places that species live and all the traits that they have into this very complex parameter and use MCMC to propose and accept and reject new possible histories. So this is, I don't want to get into too many details here, but this is kind of the magic of why this works, why we're able to do inference under it. So if you're a methods geek like I am, it's sort of interesting. But for many people, just take it for granted we did something to get around a sort of thorny inference problem. Now, probably what's more interesting is we then applied this model, an inference method, to study how Galapagos finches potentially evolved throughout the archipelago, the Galapagos archipelago, I should say, if that's not obvious. Now, for this analysis, we used a time-calibrated phylogeny by Lamashani and others from 2015, which includes 14 species using a time-calibrated phylogeny based off of sort of phylogenomic data. We used principal component transformed traits for beak shape, beak size, wing length, and tarsus length, which are thought to be important traits for resource use in the Galapagos finches. And then we also used range data for 19 regions throughout the archipelago where these species are found. Now, our framework is a Bayesian framework, so our parameter estimates are posterior distributions of parameter estimates. So we don't have point estimates. We have to think about ranges of possible parameters that are consistent with the data. And remember the three parameters I mentioned earlier correspond to three different types of hypotheses for competitive interactions or biotic interactions. First, we have omega X on the left. That's for traits. And we ran the analysis 4 times, once for each of the 4 traits. So these distributions each correspond to a different trait by color. Now for omega X, for all of these hypotheses, by the way, if the parameter value is 0, that means we don't have a strong effect one way or another. So what we're looking for are distributions that really are away from the value of 0 to say that we think something is happening. So for omega X, which corresponds to traits, in general, we found a fair amount of overlap with the 4 traits with respect to the value 0. So we basically concluded that we had mixed signals for convergence and divergence, depending on what traits you looked at. And we couldn't say that there was a consistent pattern in terms of biotic interactions in finches across for ecological traits. So we don't necessarily think character displacement is driving trait evolution in the system. But when we get to omega 1 and omega 0, which corresponds to colonization and extirpation respectively, we did find stronger consistent signals of negative omega 1 values. So that means that interactions impede colonization. It's harder to enter a region if species with similar traits are already there. They're negative and far from the value 0. We had weak and but somewhat mixed results for support, saying that extirpation is higher in regions if sympatric species have similar traits. But overall, we felt these results seem more consistent with competitive exclusion than, say, character displacement in terms of explaining patterns of biogeography and trait evolution. Now, these models also let us plot the estimated effects of interactions back onto the phylogeny. And what we have here on the left-hand side is a cloud of all the simulated trait histories that we use for that data augmentation approach. On the y-axis is beak size, a principal component for beak size. And the gray to purple gradient here is a deterministic effect of interactions on trait evolution. And the important thing here is that whenever we see purple, that means that for this trait beak size, we see some signal that species traits were deterministically driven by competitive interactions. So there's some character displacement. Now, I just want you to remember these 2 species, because I'll mention them again in a slide or 2. These 2 species, Fusca and Oliviaceae, are these 2 species here. Very similar indeed, right? I actually had a hard time being certain that Google image search was showing me the correct 2 species because they're so similar. And they've actually been a little bit of a conundrum as far as I understand in terms of finch biology. So we can also map this deterministic effect of competitive interactions on species ranges. And so this is not the prettiest figure, but how you'd interpret it is, this is a phylogeny, if you squint at it, with species relationships given by these sort of nested clades. And for each lineage, we plot the effect, the effect of competitive interactions on extirpation rates for each of the 19 regions, which are listed in rows here. And we can kind of see that overall extirpation is slightly faster for species with similar traits to others in a region. And for colonization, the effect is much stronger. So just this pinker color means that they're stronger than this lighter color for extirpation. But again, this figure is not wonderful, but I do want to show it just to give you a framework for understanding this next one, which I think is more revealing. I mentioned these 2 species 2 slides ago, the ones that look very similar. We focused on, in this result, we focused on the effect of interactions on the colonization rate for these 2 species. Now, we're just looking at them head to head for the 19 regions. Now, if the node or if the circle is colored black, it means that species occupies the region. So Fusca occupies Wolf Island and Darwin Island, but Oliviaceae doesn't. Now, if the color is pink, it means that the island has a higher probability of colonization in the next moment of time. If the island is purple, it means it has a lower probability of colonization. So look what happens. They're very similar in traits in all typical regards of morphology. if species C. fusca is in Wolf Island, it means that Oliviaceae, purple, cannot easily colonize the island. And vice versa. Here we're in Pinzon, and fusca can't colonize it. Lower colonization probability. But in the islands where neither species occupy it, each species has a higher probability of colonization. So it's very interesting that we get that this model recovers these it recovers a strong signal for effects of competitive interactions on colonization rates in such a way that it also reflects the patterns of allopatry you see between these two incredibly morphologically similar species. That's pretty interesting. Okay, so for the next, I think we're to the next part of the talk. Right, now we'll shift gears a little bit and talk about how two clades might co-evolve with one another rather than thinking about evolution within a clade. This section is focused on what are often called co-phylogenetic methods and co-phylogenetic patterns. Some of this work was pioneered in the system of pocket gophers and chewing lice, where you have highly specialized parasite taxa, the chewing lice, that are thought to basically diverge whenever their host species, the pocket gopher species. And especially because chewing lice have a very low rate of host switching between host lineages, it gives you this very strong co-phylogenetic pattern. So the topology for this clade, for example, it mirrors the topology for that little subclade. That's the basic idea. Now, in other systems, it's much messier. Actually, I'd say many systems that have not been studied as easily as the pocket gophers, it's much messier. So for example, with orchid bees and orchids in this nice paper from 2011, we see that orchid bees, which apparently carry pollen in distinct parts of their body, but also might be receptive to different types of chemicals that are put out by the orchids. You have lots of host switching. You have some orchid bees make use of multiple orchids, some orchids make use of multiple orchid bees, and so forth. Much messier. And so, the third part is that when thinking about the way in which lineages gain new abilities, gain the ability to use new hosts, there seems to be some type of relationship between that gain of use and phylogenetic distance. Namely that parasites seem to more easily gain new hosts that are closely related to hosts that they can currently use. So in the same way that geographical distance, that's what this paragraph will say, what I'm about to say. In the same way that geographical distance seems to shape the way that species disperse among regions, phylogenetic distance seems to shape the way in which parasites colonize new hosts. Pretty interesting. And biogeography and host-parasite evolution have a strong historical tradition, especially in the phylogenetics literature. And for this part of the talk, I started working with Mariana Braga and several other Swedish researchers on modeling how we might better account for the way that that parasite species gain and lose hosts, especially in this phylogenetic context. And the three questions we set out to answer were, does host phylogeny predict rates of host colonization? Can ancestral ecological networks be reconstructed? And if so, when can network structure first be detected? In this model, we have a pretty simple core. The idea is that each species can make use of some number of hosts. So for example, suppose a parasite species can make use of any host A, B, C, or D. That species might lose hosts at rate lambda 0. So let's say we lose, here in this case, we lose host B. We also might gain hosts at rate lambda 1. So in this case, we gain we gain host A at rate lambda 1. We also introduce what might look complicated, but it's actually a pretty simple function for accounting for phylogenetic distance. I just want to show you this function to say that there's this factor beta, which controls the importance of phylogenetic distance. And what this parameter beta does is it says that when beta is equal to 0, phylogenetic distance among hosts has no effect. And when beta is greater to 1, larger phylogenetic distances between hosts means the rate of host gain is slower. And as a cartoon, what we're doing is we're asking, suppose your parasite species uses hosts B and C and wants to gain host A. we're going to account for the distance from B to A and for C to A when we're thinking about that rate of host gain. That's the basic idea. What's complicated about this is that once you start talking about the rates at which hosts are gained, and its dependence on the current set of hosts you use, it means that all of your hosts must be accounted for when you're thinking about host gain rates. And this means that your state space, all the possible combinations of hosts in your model must be accounted for in some way. And if you've worked with these Markov models before, continuous time Markov chains and so forth, what this means is your Q matrix suddenly has 2 to the n possible repertoires, or it's 2 to the n possible combinations of host use that you need to model, which is a large number. For example, if you have 10 hosts, that's about 1,000 by 1,000 hosts. Now, to get around that problem, and again, I won't get into it, we used a similar data augmentation technique, as I mentioned previously, for the Finch paper. But again, I won't bore you with the methods stuff. To actually apply the model and method to an empirical example, we worked with a butterfly group, Pieridae, which include the cabbage butterflies, and their use of 50 different angiosperm families. And the data set we had basically looked like this: a time-calibrated phylogeny here, a cladogram here, and an interaction matrix that was 65 genera by 50 families. Now, when we fit the model, we found that host phylogenetic distance did influence colonization rates. The value beta was positive and away from 0. So this is the mean posterior estimate and the highest posterior density. It was definitely non-zero and positive. So we did say that that phylogenetic distance among hosts affected host gain rates. Our analysis also was able to reconstruct several interesting transitions into new sort of ecological units or modules of plant families. So for example, ancestrally, Pieridae used what we're calling module 1, the Fabaceae group, and then they transitioned into Caparaceae, the caper family, which is we're calling M2. Twice they moved into Loranthae and Santalaceae, which includes some interesting parasitic plants here and here. And then at least twice the Pierids entered Brassicaceae, the cabbage group. Now, as part of this, we were also interested in thinking not just about interaction matrices, but also what that might mean for the structure of those matrices. And when ecologists study this from the modern viewpoint, by which I mean present day data, they often use ecological networks to highlight structure in communities. So we were wondering, what would it look like to try to reconstruct that network over time? And what we did is we took time slices of stochastic mappings for these histories at 10 million year intervals. And it looked something like this when we pieced it together. When you go back to the root of Pieridae, when we have the fewest lineages, we found, of course, there are fewer lineages represented at that point in time, simply because many lineages have went extinct since then, but also because that was probably when the clade was its smallest. But we found relatively small networks with predominant dependence upon Caparaceae and Fabaceae, and some early differentiation between the Fabaceae and Caparaceae components. As we move forward into the late Eocene and through the Oligocene, we see more apparent modularity emerge and the formation of new interactions with Brassicaceae, Tropalaceae, Loranthaceae, and Santalaceae, and so forth. Once we start to get into the early and mid-Miocene, we really find we find that modularity persists, but also that the modules themselves begin to become interconnected. And by 20 million years ago, you get what appears to be the first occurrence of what's sometimes called a giant component, which means all nodes are essentially accessible by a path of any length from any other node in the network. So everything is connected is one way of putting it in short. Here's the reconstruction overall of the different time slices. And what we like about this figure is it really shows how a network kind of grows in a phylogenetic sense over time, which to our knowledge hadn't been done before. Now, the networks themselves have some interesting properties. We also used our Bayesian framework to test and try to detect when first aspects of network structure appeared back in time. And to do this, we looked at we basically, we computed modularity scores and nestedness scores with respect to time, and asked, when does our posterior distribution of stochastic mappings first exclude the zero values, so the non-nested and the non-modular network scores? And in both cases, we found that by about 30 million years ago, we're pretty confident that modularity and nestedness occurred. Now, it could be that those features existed before, but given that we find extant modularity today and extant nestedness today, it seems likely that that these are real signatures of at least an early detectable signal for those network structure features, if it's not the first. I mean, those features could have arisen 60 million years for all I know. But this is at least the earliest one that we've been able to obtain to date. So we'll see where this research goes in the future. Hopefully somebody will find fossil evidence of, caterpillars feasting on leaves that we can incorporate into these models and improve the estimates further. Now for the last component, and I'm becoming aware I'm running out of time, it'll focus on how that plant clade I mentioned earlier, viburnum, interacts with its environment, and in particular major habitat types or biomes. Here's a picture of some biomes displaying some really lovely red fruits in a freezing winter. Now, people who work in deeper phylogenetic scales and are thinking about ecological interactions often have to simplify what those ecological interactions look like into simple statements about major habitat types, which it's definitely dissatisfying on some level, but it can give you some insight into what's happened over deep timescales for these really fascinating clades. So in this group, Hakea, which is part of Proteaceae in Australia, here's Australia. Oh, and here's Hakea. This group of plants occurs in a number of habitat types, many of which surprisingly are Mediterranean adapted, this light blue color. So here we have the phylogeny. The tips are color coded by the percentage of occurrence records in each biome, which have been classified as such. And many extant taxa are Mediterranean. They recover a Mediterranean ancestry. And this type of reconstruction can help you identify when and potentially where did these transitions between different habitat types occur. Now, depending on the time scale and the regions you're looking at, the entire endeavor is straightforward or a little bit complicated. So for example, if you're looking at extremely deep time scales and you're looking at a global scale, you might be reluctant to say that you know exactly what the distribution of biomes is at any given time. And the distribution of biomes certainly should affect how easy it is for different species to transition into different biomes, depending on where they live. So here's a nice review paper by McElwain in 2014, which shows, wow, almost 500 million years of plant evolution, how that distribution of vegetation across the Earth has basically defined what biomes we have in different regions at different time points. So for example, these colors aren't easy for you to see, but if you're interested in thinking about the evolution of tropical everwet plants, if the lineage is, say, 150 million years old, you might want to take into account the fact that the Earth was much wetter and much more tropical throughout, positive to negative 30 degrees latitude 150 million years ago. That might be relevant. If you're interested in modeling evolution of species in grasslands, you might be interested in the fact that grasses are not particularly widespread and dominant as a biome until, say, the Miocene, or perhaps the Oligocene. They seem like relevant factors for these types of questions of habitat evolution and reconstruction. So what we were doing in this viburnum paper was thinking about how the distribution of biomes might potentially change the geographical opportunity for dispersal and also the geographical opportunity for shifting between biomes depending on the ages of different lineages. And also, just to make it complicated, where lineages lived in the past. So I'll try to piece this together for you in the next few slides. And this work was described in 2 sister papers that came out in the same issue of Systematic Biology. The paper I'm going to focus on is this one here, Modeling Phylogenetic Biome Shifts. And this is more of a modeling problem than the other one, in my opinion. And the questions we are grappling with are, how can we encode paleobiome structure? How do we inform phylogenetic models of paleobiome structure? And does paleobiome structure have an effect on ancestral biome estimates? Just as a cartoon, the sort of challenge here is, suppose I have the same phylogeny. So it's the exact same phylogeny on the left and on the right. And I've also coded the species with the exact same habitat use characters. So cold, warm, cold, hot, warm, cold, warm, cold, hot, warm. The reconstructions are very different, but they both give rise to the exact same distribution of habitat affinities. And so the question is, which of these 2 histories is more probable? And this one on the left prefers a warm ancestry. This one on the right prefers a cold ancestry. So it depends to some degree, I would think, on what were the ancestral conditions of the planet. So how can we bring that information into the problem? Now, this study is really designed around viburnum specifically, so I'm going to show you more about viburnum briefly here in this slide to give context to the rest of the talk. So viburnum has about 165 living species, primarily distributed throughout the northern hemisphere. I would say about half of the species, are found in cold temperate habitats. About 30% are in warm, I would say about 40% of species are found in warm temperate or cold temperate habitats. For this talk, I'm sorry, I misspoke. Let me start over. About 50% of species are found in cold temperate habitats, the dark blue. About 40% of species are found in either warm temperate or cloud forests, which is the light blue. And the remaining roughly 10% of species are found in tropical forests, colored in red here. And one thing that's pretty striking about this distribution of viburnum species with respect to regions is that you certainly don't find many cold temperate species in Southeast Asia, nor do you find many tropical species in North America. Today, that might not be surprising, but the distribution of biomes hasn't always been this way, right? But clearly, just looking at extant distributions, it makes sense. So let's just look at a few different time slices to see how this might change your perspective on where we find different species with different biome affinities. So here's a rough sketch of where tropical biomes are distributed today in red. What we're going to do in this model is take this information, which is hard to obtain in the past. So let me give that qualifier. It's hard to obtain for paleo reconstruction. So what we're going to do is take distributions like this, information of this type, and try to encode it into an adjacency matrix. And what this adjacency matrix does is it shows which regions have strong, weak, or marginal features, such as biome availability on the diagonal, or connectivity on the off diagonals between regions. So here we have South America at present. It has strong availability of tropical biomes, and it has strong connectivity of tropical biomes with Central America. But even though Southeast Asia and South America both have tropical biomes, they do not have strong connectivity. Okay, that's the basic idea. So that's what the present day looks like. We can do something similar for the mid to late Eocene, where tropical biomes were much more prevalent. And you can see the short of it is you can see more cells in the adjacency matrix are filled in. Greater connectivity, greater prevalence of tropical biomes. So that's the basic information we're going to work with to inform our model. Now, I said it's not easy to obtain this information, so we relied on several papers that gave qualitative descriptions of paleo biome structure for 8 epochs, 6 regions, the ones where I described viburnum previously, and 3 mesic forest habitat types, or 3 biomes, which are tropical forests, warm temperate forests. Now these include the cloud forest lineages of Central and South America, and cold temperate forests, which are biomes that regularly experience long periods of freezing during winter months in particular. Now here, time is on the y-axis, the major y-axis. And here let's look at the period of time between 100 million years ago and let's say towards the late Eocene at 33.9 million years ago. And we have, in general, high degree of availability of tropical biomes, a good amount of connectivity, and a strong representation of warm temperate biomes and connectivity, especially in the Northern Hemisphere. But essentially little to no dominance of cold temperate biomes or connectivity of cold temperate biomes. This seems to be a real feature of the paleobotanical record. You just don't find cold temperate forests abundant on Earth when you go back before the Oligocene. Now let's look after the Oligocene, or beginning with the Oligocene and further. Beginning at 34 million years ago, that's when you see the rise of what we now call the boreal forests. The tropical forests begin to fragment. The warm temperate corridors of the boreal tropical forests also dissipate. And when you reach the present, this is what the world looks like today. Isolated tropical biomes in different continents, weak connectivity between the Americas and the old world, Southeast Asia, East Asia, and Eurasia and Europe, and some amount of connectivity in the Northern Hemisphere between North America and Eurasia, but not much. These are major features that are described in the paleobotanical literature, like I said. The short of it is that the boreotropics are much hotter and wetter before the Oligocene and cooler and drier afterwards. So those are the 2 major features you need to think about moving forward here. Now, in terms of the phylogenetic model, now we want to think about how could these lineages evolve within this context. So to handle this, what I'm going to do is define 18 compound states, which are combinations of regions and biomes. So I've color-coded these states such that the color or the hue gives the region, and sort of the lightness or darkness gives the biome or habitat type. So light colors are tropical, dark colors are cold. Now, in this biome shift, in this model that I'm going to define, we'll use a continuous time Markov model. And what's important to think about here is the rate matrix model that will apply to it. So this rate matrix will have a block matrix structure that looks like this. So the block diagonal matrices give us dispersal rates between regions, and the off diagonals let us define rates of shifts between biomes depending on what region a lineage is in. Here's what the structure looks like when we actually populate it with rates for an example from the mid to late Eocene. And what we can see is that, for example, if you are a worm adapted lineage, no matter what region you're in, it's very difficult to transition into cold temperate environments because they just didn't really exist at that point in time. It's possible, but the rate is lower. Okay. Also, if you're a worm adapted lineage, it's fairly easy to disperse between East Asia, Europe, and North America relative to other areas at that time period, based on what worm biomes were present in what regions at that time. Now, I just showed you one time slice, but we're actually going to construct matrices like this for all time slices. And I don't have another slide, but one of the tricks of this model is I'm not really hard coding exactly what these rates are. So I'm going to estimate these rates in a sort of an empirically informed manner, but the model still allows me to estimate weak effects, no effects, ambiguous effects, and so forth between regions and biomes. So it's not as strong as these images appear. The assumptions are not as strong as they might appear. Now for these results, which I'm going to have to blaze through, we constructed 3 different frameworks to sort of suss out how paleobiome structure may or may not matter in terms of modeling habitat evolution. So the three frameworks we used are first this paleobiome framework. For this, we're going to use the full set of features for how regions and biomes are distributed on the planet over time. The second setup was what we're calling a modern biome analysis. And for this, we just imagined that the distribution of biomes like so, where you have fragmented tropical forests and prominent boreal forests, cold temperate forests rather, in the northern hemisphere. That's what it always looked like throughout the past. And then finally, we used a biome structure called the null biome structure, which essentially ignores space and time. It says all biomes are present at all time periods. Don't worry about it. Let's just get on with the inference, which is probably I should mention, this null biome style of analysis is most similar to what is commonly done in phylogenetics today. So we're just trying to improve beyond that, push the needle a little bit. Now, if we do ancestral state estimates or ancestral biome region estimates rather, under these three different biome structures, here's what we recover. I just wanted to show you the whole phylogeny to give you a general sense of what the distributions look like. There's no way you can see those details, but focus on the root state estimate here. This is probably the most interesting node of the tree. The three biome structures recover similar regions of ancestry. They're all East Asian, 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 and so on. But the three different models reconstruct different habitat preferences depending on what biome structure you assume for the inference. So if you use the paleobiome structure that's informed by the paleobotanical record, the reconstruction strongly prefers a warm or tropical, but certainly not a cold temperate origin. If you only use modern biome structure, which is reflective of a much colder climate than most of history for the past 100 million years, you reconstruct, guess what, a cold temperate origin. And then the null biome reconstruction, in some ways, because it's so ambiguous, it kind of saves you from accidentally reconstructing something that's probably unrealistic, a cold temperate origin. But it still gives quite a bit of credence to that possibility. Almost 1/3 of the probability still allows for cold temperate origin. If you look at the distribution or the proportions of lineages that are in these different lineage states over time, for the paleobiome model or the paleobiome structure, you see that for, let's say, the first 40 million years, you have a very large representation, an excess almost of lineages in warm, temperate, and East Asian states. But when you use the modern biome structure, that signal almost entirely disappears. And cold temperate lineages become the dominant component of the ancestry of the group, all the way up until the Oligocene. I didn't show the null biome reconstruction because it's very similar, but it's almost identical. So that's basically the talk. Again, I focused on three different layers of modeling ecological interactions. You might want to do it within clades, between clades, or among biomes. Here's where I start to roll out all the asterisks as to why this is difficult. Oh, not quite yet. Let me just briefly summarize what I showed you. Again, within a clade, I was looking at Darwin's finches and how competitive exclusion might help shape patterns of allopatry. Between Pieridae and angiosperm host plants, I was looking at how one might reconstruct ecological network structure. And then in Viburnum, I was modeling how those species might disperse and shift between different biome types over the past 65 million years. Now, there aren't very many phylogenetic models of ecology because it's not terribly easy to do for a large number of reasons. And by no means am I saying my models are particularly good, but it's difficult. First and foremost, from the modeling perspective, you just cannot use standard likelihood and inference machinery to explore these types of processes in a statistical framework. It's too bad. I wish we could use Felsenstein's pruning algorithm for everything, because then it would just be a solved problem. And then it's just a matter of defining the model and applying your imagination and your scientific rigor to getting it to the bottom of things. But there is this sort of fundamental computational difficulty to inference. There's also a statistical problem, which is that the models are extremely prone to over-parameterization. There is a large number of possible factors that could influence how lineages evolve in an ecological context. But we can't simply add a parameter for every possible condition. Another limiting factor is that biological data is extremely finite. It's hard to obtain. It doesn't always preserve well. It can be noisy and so on, which means that it's hard. You can't really develop a custom model for ecological evolution that will solve all problems. It seems like you can do that for some types of molecular data, but maybe not for ecological problems. For me, I'm not sure if all modelers see it this way, but for me, I think that designing detailed models really requires expertise across domains, and it invariably relies on strong collaborations with people whose opinions and insights and familiarity with the literature you can trust. And you know, I think that these models are such a wonderful bed for developing frameworks where the biology and the modeling folks really come to the table together and agree on what's important and what's not. For me, I just found it incredibly fruitful and enriching. Now, there are some methods that others have been developing that's kind of skirt around at least the technical issues that I just mentioned. And these are often in a simulation-based framework. And the idea is that you can apply first principle reasoning and use models to simulate or generate data sets, and then use non-parametric modeling frameworks or inference methods to essentially detect patterns in these simulated data sets. And I think there's a lot of promise in using these types of approaches that hasn't been explored yet. But these are two really wonderful papers that have come out recently looking at similar types of problems, but from different timescales and making use of different types of data. So I would check those out if you're curious about this. This probably is my last slide. I'm sorry, I'm at the end here. But basically, I wanted to end on an upbeat note. I really just want to emphasize there's so many wonderful things in developing non-standard models, especially for ecological problems. I mean, it's really rewarding to try to connect these models back to the biology and to the literature and the theory. It leads to so many enriching conversations and collaborations. All of the work that I've shown so far depended on working with people who really know the organisms, really think about the details of the process, and aren't just trying to write down models for which you can compute tractable likelihoods. It's a little bit more engaging when you realize that the models are failing the biologists, and how can you actually do something productive for the whole community. I think that working on these types of models also really strengthens your ability to conduct hypothesis-driven research, because at some point you have to include or exclude certain hypotheses from being covered from the cases your model studies. And it's interesting to think about how your models may or may not be sensitive to those blind spots. If you're only caring about one type of hypothesis, you might be blind to all the other explanations for the data. Some of these are redundant. I think this is kind of redundant. I'll skip that. And then finally, it's incredibly humbling. You realize how difficult it is to develop models in general. And no matter what, you should just really appreciate how much work people put into these things. It's incredibly difficult to even make small steps of progress. So that's basically all I had to share. Again, I just want to thank Anya for being such a wonderful host and for giving me the opportunity to speak with everybody. I've had a lot of fun meeting everybody, and I hope if anybody has questions about this work, you feel welcome to contact me in any way you feel comfortable. The last thing is I just want to point out Ignacio Quintero and Mariana are postdocs, and they're both awesome. So if you're looking for someone to give a talk or know of job opportunities, let me know and I'll put you in touch with either of them. Thank you.